Is just nice to be here with sunshine. I'm in California, and the Mind Institute, which is actually pictured here, let me get it on the presentation thing. Here's the Mind Institute, and I actually came here in 2000 after spending 20 years at University of Colorado. It was nice to come back to California because I was born and raised in Berkeley. And it was also very nice to come back to a place that had an whole institute focused on neurodevelopmental disorders, because at University of Colorado, you know, we were the kind of money-losing part of the medical center and relegated to an old rehab location. And in fact, one of my fellows had her office in the old hot tub room toilet area. So, you know, going from those digs to a beautiful new institute that when you walk in the front door, it was designed by families that had children with autism, and they wanted families to feel a sense of hope as they walked through the door. So it is actually a beautiful institute, and it's been a joy to be there since 2000. Well, I want to get you excited today about the association between Fragile X and autism. I'm going to talk about a whole range of disorders related to the Fragile X gene that's on the bottom end of the X chromosome. I'm going to talk about multimodality interventions, but I'm also going to talk about the diagnostic process, which needs to really have multiple professionals involved. Oftentimes, a therapist who has worked with a child with Fragile X, because of the very characteristic phenotype of poor eye contact and hand flapping, sometimes hand biting, anxiety, and hyperarousal, will recognize it when they see it again, and will recommend to the family that this kid should be tested for Fragile X because of the similarities in the behavioral phenotype. And sometimes the physician may not have even heard of Fragile X. We oftentimes hear that, or Fragile X-associated disorders, which we'll be talking more about. In fact, I've talked to several families here today that have taken their children or adult individuals to multiple professionals, and nobody tested them for Fragile X. So we want to change that here. I'm going to start out with some general comments about autism. And everybody's seen the DSM-IV diagnostic criteria for autism, but I want to point out that autism is a behavioral diagnosis. It's not a medical etiological diagnosis. So it's like attention deficit hyperactivity disorder. There's many, many causes of this behavioral phenotype, and so you have to look for the causes. Same with autism. So I'm going to talk a little bit about the diagnostic workup, because everybody's very familiar with the diagnosis of autism, but sometimes this diagnosis is made often by a psychologist who's doing standardized testing to make the diagnosis, and Kathy Lord and other individuals have made it much easier with standardized testing to make the diagnosis, but you have to work with the medical people, pediatrics, psychiatry, neurology, who are ordering the blood workup. You know, you have to do blood tests to find out about the specific cause of their autism. There's not a single explanation for autism. It's a behavioral construct. You know, we're able to figure out the cause now in about 20 to 30, and some papers even 40% of the causes of autism. So you have to look for that. This is definitely a multi-professional endeavor. And I'm going to just talk about that workup. I think that the advances in molecular biology and neurobiology, and also in brain imaging, have really taught us a lot about what is going wrong from a variety of different known etiologies. And of course, we're always investigating, you know, whether there can be additional environmental toxins that could be adding to a genetic vulnerability to autism. And I think that research is actually in its infancy and will definitely be expanding. And we're always interested in how to improve educational and therapeutic endeavors for individuals with autism, and starting early is absolutely essential. But what we've learned is that there are multiple genetic loci. Some of the newer studies with copy number variants are showing us that there are abnormalities in different parts of the genome, and maybe additive genetic vulnerabilities that can lead to autism. That there can be a very broad spectrum. I mean, on a regular basis, I see kids that are gifted with autism. I see kids that are severely retarded with autism. I see kids with multiple, physical anomalies with autism, and kids that look absolutely normal with autism. So it comes in all different varieties. Some of the commonalities, particularly with functional MRI studies, show abnormal processing and abnormal connectivity in different parts of the brain. In fact, I just read a paper that came out in PLOS this week that talked about white matter abnormalities in 48% of individuals with idiopathic autism. So even the neuroimaging is changing, looking at abnormalities in white matter that when you use the right kind of T1, T2, and FLAIR imaging, that you can demonstrate some of these abnormalities that many studies have not seen before. We do know that there are a variety of genes that control the protein expression at the synapse, where two nerve cells come together. And if there's mutations in any one of these genes, it can cause abnormalities in synaptic connectivity. And most of these genes, when mutated, can cause an autism-like phenotype. So we'll talk a little bit about that. So anything that interferes with synaptic plasticity, could be related to autism. We also know that GABA systems in the brain, these are inhibitory systems, and glutamate systems, stimulatory systems. Sometimes these systems can develop an imbalance, and there's a variety of genetic mutations that can cause gamma and glutamate abnormalities in the brain, and many of these gene mutations are associated with autism. So many of these neuromodulatory functions of GABA, glutamate systems, synaptic plasticity, etc., can affect how different parts of the brain connect. And that interference in connectivity can lead to different forms of autism. So some of the themes are going to be synaptic plasticity abnormalities, excitation and inhibition abnormalities, connectivity problems, and neuromodulation problems. All of these problems can be associated with autism. We do know about structures that seem to be more involved in autism than other structures. The prefrontal cortex, the amygdala, this red area here, really important for the processing of emotional information, eye contact, that sort of thing. The somatosensory cortex, and many of these individuals can under or overreact to various types of sensory stimuli, and the insula that helps to integrate all this information and connect to the emotional or the limbic system of the brain. These are areas that are often dysfunctional. There's been some new research about neuroinflammation in autism, and people are very interested in autoimmune problems that could relate to autism, and we will be touching on that, particularly because there can be autoimmune problems related to Fragile X. Altered brain serotonin levels, and serotonin is a very important neurotransmitter both for the limbic system, the amygdala, hippocampus, also throughout the frontal regions of the brain. And there's evidence that there can be low levels of serotonin early on in childhood in these regions of the brain, and serotonin is kind of trophic, helps to stimulate neuronal connectivity and growth. And so there's a lot of research going on now about starting selective serotonin reuptake inhibitors, or SSRIs, very early on in autism to stimulate language, decrease obsessive compulsive behavior, and also stimulate social interactions. So that research is pretty exciting too. This is from a slide by Belmonte and Bergeron talking about many pathways that lead to autism. Some of the things that I've been talking about, we're going to talk a lot about FMR1 gene dysfunction, the mGlu-R5 system, and this postsynaptic density protein that's important for synaptic plasticity. MECP2 is the gene that is mutated in Rett syndrome, which can cause an autistic-like phenotype. These are tuberous sclerosis genes, neurofibromatosis. P10 is a gene that regulates growth, and when it's mutated, it can cause very large heads that can be seen in a significant number in one study by Merlin Butler, in 18% of individuals with autism with big heads. Chromosome 15 duplications can be associated with autism. Neuroligin mutations and also neurorexin mutations, which are associated with synaptogenesis, the connecting of synapses, as I talked about. These can also cause autism. So there is a variety of structural changes that can happen in the brain related to all of these different mutations, and this can lead to the susceptibility of autism spectrum disorders. There are whole lists of genes, when mutated, which can cause autism. This is just one of these lists here, but the list has grown over the years, and some very exciting new genes have come out lately. So for the clinician that is doing the blood work and the workup of individuals with autism, and again, this may be after the psychologist has made the diagnosis with standardized quantitative new measures like the ADOS, the ADI, and other measures. I think that behavioral evaluation of autism is really important, but just as important, I think, is the medical workup for autism. And Schaefer and Lutz have published papers, even more recent papers, that have demonstrated, in this particular paper, 41% of the individuals with autism, they were able to find a specific genetic disorder. So there are not only high-resolution chromosome testing with FISH studies, and of course Fragile X DNA testing, which we'll talk more about, metabolic studies. You can also do gene sequences. But actually, this number may be even more enhanced with these new, what we call CGH arrays. These are studies, stands for comparative genomic hybridization. We now have probes for the whole genome, and these probes are getting more and more and more detailed. So now, really, the workup of autism, we think that CGH arrays are medically indicated because they can pick up subtle deletions, subtle duplications, variations in different parts of the genome. And just with this test alone, it can detect, somewhere between 20 to 30% of patients with autism that can have a genetic abnormality. So there are several centers that have great expertise in doing CGH arrays. Baylor is one of the centers that we routinely send to, but there are a lot of private labs that are now doing CGH arrays for making the diagnosis of autism or looking at the genetic cause of autism. Again, the diagnosis is made behaviorally, but the genetic workup is absolutely essential. So I want to propose that Fragile X is really a great model for autism because many children with Fragile X have full autism by ADOS, ADI diagnosis, about 30%. And I apologize. whenever I give a lecture and they want the slides 2 weeks ahead of time, I'm usually traveling or giving another lecture, so I don't put enough time into what I send 2 weeks ahead, and really most of the time happens the night before in fine-tuning the lecture. And I gave a lecture yesterday, and I got all kinds of input about what I should talk about today. So I apologize. Your handout is, what I was thinking about 2 weeks ago, and things dynamically change, but usually for the better. So you have most of my slides, but not all, or they might be out of order, and so I apologize for that too. But believe me, what I put together last night is much better than what I put together 2 weeks ago. So I think Fragile X is a great model for autism. 30% of children with Fragile X syndrome have full autism. Another 20 to 30% have PDDNOS, or are on the autism spectrum. So really, we're talking about 50% plus that have an autism spectrum disorder. And then the ones that don't meet full criteria for autism or even PDDNOS oftentimes have poor eye contact, hand flapping, and a lot of autistic-like features, even though they might be very interested in social interaction. So autism is really very tightly interrelated. Fragile X, I think, is the most common genetic cause of autism. But if you take 100 children diagnosed with idiopathic autism and do Fragile X DNA testing on all of them, you will find anywhere from 2 to 6% that come out with a Fragile X mutation. And I'll talk more about the full mutation and the premutation. Both disorders, Fragile X and autism, have big heads, rapid brain growth early in childhood, and Fragile X with autism has a bigger head than Fragile X without autism. Fragile X has problems with hyperarousal and anxiety, as I will show you, and I think a subgroup of idiopathic autism can also have very similar problems. The reason it's very important to sort out the subgroups of idiopathic autism that have a similar phenotype to Fragile X is because there is now targeted treatments for Fragile X that reverse the neurobiological abnormalities. So I think Fragile X is leading the way in terms of new targeted treatments that will reverse the mental retardation in autism. And some of these treatments, because of the molecular overlap with other causes of autism, some of these new treatments that I'm going to talk to you about today, I think will work for idiopathic autism that doesn't have the Fragile X mutation but has similar neurobiological pathways. So I apologize for all of the neurobiology you're going to hear about today, but for me, this is really an exciting time. And I have been in this field for actually far more than 20 years, and this is the most exciting year, and I'll hope to generate some of that excitement to you. So for all those kids out there that didn't want to be tested for any genetic thing that could be causing their autism, I think the tables will be turned now, because if there's a targeted treatment that can reverse the abnormalities, they want to be on the bandwagon to get the treatment. So I think this will really create an upswing in how many individuals get genetically tested. Both disorders have problems with eye contact. Fragile X patients tend to actively avoid eye contact. It actually hyperarouses. Direct eye contact overactivates the amygdala, this emotional area in the brain that's really important for coordinating eye contact, and in other causes of autism that can be the case too. We'll talk a little bit about the AMPA receptors, which when there's a lot more AMPA receptors, there's strengthened synaptic connections. And those with Fragile X and autism together really have a lower IQ than Fragile X alone. The level of the amount of Fragile X protein does not really correlate with whether they have autism or not once you control for the IQ. So with Fragile X and autism together, there seems to be an additive effect or an additive degree of dysfunction. Now, FMRP, the protein that is made by the Fragile X gene, is one of these mother proteins that controls a lot of other things. It actually hooks up to messenger RNAs from many other genes, and it controls how those messages, those RNA messages, gets converted into proteins. So it is kind of a control protein for many other systems in the central nervous system, which we'll talk about more today. I wanted to point out, this is some data that we published last year on the ADOS. This is on the old scoring of the ADOS. Each one of these triangles here represents an individual patient with Fragile X syndrome, and this is the communication and social total score here, and with this module there's a cutoff for autism here. So you can see it's really a continuum how significantly affected individuals with Fragile X are in the communication and social aspects. And this is the repetitive behaviors are pretty similar whether they have autism or not. But with the new scoring that came out where the repetitive and perseverative behaviors are pushed into the diagnostic criteria, it actually shifted about an additional 30% of individuals with Fragile X from on non-autism spectrum into the autism spectrum. Occasional individuals shifted out, but many more shifted in now that the repetitive and perseverative behaviors are taken in. I also want to point out that individuals with Fragile X overreact to sensory stimuli. And that's really important for you teachers in the audience. Here's a guy, an adult actually with Fragile X, in the photography studio that is giving me some nonverbal signals that he has just about had it with the number of pictures I'm taking of him. So he's getting overstimulated here, and you as a teacher, you don't want to start, raising your voice to him when you see this nonverbal signal, because you will likely be hit. Okay, you want to launch into some calming techniques, and basically Lucy Miller, who's a great OT person in Denver, she did some of the early electrodermal responses comparing normals with Fragile X, and these represent the sweat responses, which are sympathetic responses, autonomic sympathetic responses to sensory stimuli. And the sensory stimuli are these lines here. So whether it's visual, auditory, tactile, it doesn't make any difference. If you give them repetitive stimuli, normal individuals habituate to the stimuli over time, but Fragile X patients do not. In fact, they have enhanced amplitude, an enhanced number of responses, and they don't habituate. So you have got to take into consideration what is going on in the autonomic system of that child with Fragile X. Now, this can happen, I think, in some other causes of autism too, but this is why sensory integration techniques are so important for calming down sensory hyperarousal. Because if they're hyperaroused, and this oftentimes goes along with anxiety, whenever the sympathetic system really puts out, you get a lot of anxiety, and learning just goes down the tubes. And so we do a lot from the medical perspective about treating anxiety, like with selective serotonin reuptake inhibitors, Prozac, Zoloft. A lot of those medications can be very important. Mood stabilizers can be very important. We really try to avoid major outbursts in the classroom. We try to really help them not have aggressive episodes in the classroom, but this really does relate to their sensory hyperarousal, which is a big problem. I was at a Cold Spring Harbor conference last week where an individual was talking to me about certain receptors that are modulated. These auditory receptors are modulated by the FMR1 protein. So in Fragile X, the FMRP, or the Fragile X protein, is missing. So they can't modulate these auditory receptors, and they get the same response whether a sound is at 60 hertz or 600 hertz, okay? It is overwhelming. So some of the Fragile X kids that you see that are like this when an alarm goes off in school, it's because they can't down modulate these auditory receptors, and they just, you know, overreact, sometimes even to quieter sounds. So kids with Fragile X are usually hyperactive, particularly the boys, but also many girls with Fragile X. Some of the physical features can include ear cupping, as you see here, but there's a lot of kids with Fragile X that don't have the ear cupping. This little girl gets into a lot of trouble. She lives in Australia. But all of these features, you know, kids with Fragile X tend to do power salutes. You know, when my kids were babies and in their high chair, and when they got upset, they do I called it a power salute, you know, because, you know, this is from the 1970s and 1980s. And you know, my kids with power salutes, you know, that is part of normal development, but it kind of goes away after 2 or 3 years of age. Well, you know, I see power salutes from individuals with Fragile X at all ages, even into adulthood. Hand flapping, seen in the majority, hand biting, you know, poor eye contact. Here are 2 brothers. This one has full autism, and this one doesn't meet criteria for autism, but as you can see, he's very eye avoidant. So these kinds of problems are very common. Perseverative speech and behavior in almost all routines. And tactile defensiveness, overreacting to tactile, you know, direct touch. This is a problem. So the features of Fragile X, they're very hyperactive, impulsive, short attention span. Those individuals with Fragile X who have a normal IQ, and many girls with the full mutation have a normal IQ. 25%, you know, of girls with a full mutation of Fragile X are intellectually impaired, but the majority of girls with the full mutation are in the borderline range. And then we see, you know, 30% or so of girls with the full mutation that are in the normal IQ range, but often have executive function, problems with planning, inhibition, tangential speech, perseveration. They overreact to stimuli, not only in the electrodermal response, but they have an enhanced cortisol release to stress. And actually, we're seeing the same things in carriers, okay? So oftentimes moms, you know, who are carriers of Fragile X with a premutation can also experience a lot of anxiety and enhanced stress. Now, we used to think this was just from raising kids with Fragile X, which is enough to cause a lot of stress, but they physiologically have an enhanced stress response. And we'll talk about some of the other difficulties that can occur in the premutation in a minute. But anxiety is real common in both premutation and full mutation individuals. So this anxiety and autonomic hyperresponsiveness, you know, kind of go together. And we've actually seen a nice correlation between anxiety measures and the severity of the autism. Now, mood instability is another problem. This can be a problem for carriers, can be a problem for normal IQ full mutation individuals, and a problem for even those with intellectual impairment with the full mutation. And oftentimes these individuals can go to mental health centers and you know, get diagnosed with bipolar disorder or episodic dyscontrol or whatever, and usually never tested for Fragile X. So I want to encourage mental health centers to also, you know, send the person to the lab, particularly if there's a family history of autism or intellectual impairment or some of these other problems that I'm going to be talking about, to see if this person has a Fragile X mutation, pre or full. So this little girl, and I'm sorry I don't have the video. My computer was stolen in Guatemala, so there went the video. But she presented just with tantrum behavior all day long, but you don't want to see a video of someone just tantruming. It's just overwhelming. So an anxiety is a real key feature. We focus on it more with females because they sometimes present with selective mutism. Often the aggression can be related to anxiety. Social avoidance can be related to the anxiety. And this has to do with sympathetic, and I see there's a misspelling here, hyperarousal. We're also going to talk about some of the GABA underactivity in Fragile X. Many GABA genes are underactive. Amygdala overactivity and overconnectivity is related to this. So this video should work. This is an adult guy with Fragile X, and I'm sorry we don't have the audio, but the audio isn't much because the audio is just me talking. And this guy has selective mutism. As you can see, he's nodding and I'm asking him questions. I'm getting up there pretty close because I can't get him to respond. He has the full mutation for Fragile X, presented with selective mutism. So we typically see this more in girls, but it's also something that we're seeing more frequently in boys now too. But usually younger Fragile X kids are so hyperreactive or hyperactive actually that oftentimes that kind of overwhelms selective mutism, whereas girls with the full mutation are often shy, socially anxious, and you see much more frequent obvious selective mutism. So Fragile X occurs in all racial, all ethnic groups, all ages. Typically, you know, if you have a really good early intervention program, Fragile X is diagnosed, you know, between 2 and 3 years of age. I just don't know why it's not. There we go. Changing here. But other places, I mean, we've seen kids not diagnosed until 5 or 6 or 11, or, you know, sometimes a high-functioning guy who, you know, went through mental health centers and got diagnosed with Asperger's syndrome or autism spectrum. Nobody did DNA testing, may not be diagnosed until adulthood. So it can happen at all ages. Other features that we see, strabismus in about 20%, and this is real subtle. We diagnosed him with Fragile X when he came and saw us at age 7, and already he has a very slight esotropia here, and so I sent him to the ophthalmologist, and he already had amblyopia. So it's important that these kids get an ophthalmological or optometric evaluation definitely before 4 years of age, if not sooner. They oftentimes can have double-jointed thumbs, hyperextensible finger joints, and big testicles is part of the phenotype of Fragile X. But the testicle typically doesn't become big, and the top 3 lines here are Fragile X testicles compared to the bottom 3 lines are normal range testicles. As you see, children develop larger testicles after age 8 or 9. So you don't see big testicles typically in the young kids, but they really get to 2 to 3 times normal size in the early part of puberty, stabilized by the end of puberty. So pediatricians are measuring testicles, but psychiatrists, never. Nope, not going to do that. You know, I encourage measuring testicles, but I don't know. It falls on deaf ears. But even some premutation carriers, particularly in the upper end of the premutation range, can have slightly large testicles. And it's actually a biomarker for really how low your FMRP is. Now, individuals that have a double hit, that is Fragile X and something else, are much more likely to have autism. So here we have a family of 4 kids here, and this little boy was born with Down syndrome. But then this boy here is a normal IQ, high-functioning boy who was very hyperactive and mood unstable and hand-flapped. So somebody did Fragile X testing on him, and he was diagnosed as Fragile X syndrome, you know, when he was 5 or 6. And so once he has a full mutation, we know that the mother is always the carrier in someone that has a full mutation. If someone has a premutation, and it's a girl, the premutation could have come from her father, but it only passes to a full mutation when it is passed on by the mother. So that means the mother's a carrier. She has 2 X chromosomes. Each child could either get 1 X or the other. And so we tested everybody, and guess what? This little girl who had shyness, social anxiety, and math problems in school, but normal IQ, turned out to have the full mutation. And guess what? This little boy with Down syndrome and really bad autism and completely nonverbal as he got older, turned out to have Down syndrome and Fragile X. And that's a rough combo. And almost always leads to autism. But individuals with birth trauma, cerebral palsy and Fragile X, or seizures, which can occur sometimes in over 20% of the males, and this gentleman here with Fragile X and autism and seizures, you can see these spike wave discharges on his EEG. He had a vagal nerve stimulator, and actually that helped his autism, helped his seizures, and helped his Fragile X. So this was a study that we published just showing that those with Fragile X plus autism have a much higher rate of seizures and combined additional medical problems than those with Fragile X without autism. This is a really interesting example of molecular overlap. So here are 2 Fragile X brothers. This one has what we call the Prader-Willi phenotype of Fragile X syndrome. So he has obesity and overeating and lack of satiation after a meal, actually small genitalia, delayed puberty. And these are a lot of features of Prader-Willi syndrome related to a deletion on chromosome 15. But he doesn't have that deletion. He has Fragile X syndrome. And FMRP is right here, and it interacts with another protein called CFIP. That stands for cytoplasmic interacting FMR1 protein. So it impacts a lot of other areas. It impacts the actin fibers in the neuron. It interacts with actually RAC and Rho, which are very important for synaptic plasticity and growth of the synaptic connections. And when CFIP was found, this is a protein that comes from a gene that's on chromosome 15 in the Prader-Willi region. When CFIP was identified to come from that 15q region, we measured CFIP expression and protein levels in patients with the Prader-Willi phenotype of Fragile X, and it was found to be very low, okay? So in other individuals with Fragile X syndrome, CFIP is very high because FMRP controls CFIP expression. So when FMRP is not there, the inhibition of that expression is relieved and CFIP is upregulated. That's in regular Fragile X syndrome, but it is dramatically downregulated in those that have the Prader-Willi phenotype. We don't know why it's downregulated. Less than 10% of Fragile X patients have this Prader-Willi phenotype, but the interesting thing is if you have this Prader-Willi phenotype, you are much more likely to have autism or autism spectrum disorder. So it's like an additive genetic effect. Here is chromosome 15. And for those of you that do research in autism, you know that this is a very hot region because when it's duplicated, it is very likely to cause autism and a very specific, kind of phenotype of autism. And here's CFIP right up here. There are other GABA receptors in this region and other very important genes. So there may be a variety of different reasons why there's more autism in this group. So recently a mouse model with CFIP deleted was developed by Joe Buxbaum, an autism genetic researcher, and he presented at the last human genetics meeting that this mouse has a lot of social deficits and autism features. So that's not surprising to us, because when we see CFIP downregulation in Fragile X with the Prader-Willi phenotype, we see much more autism. So we definitely want to do more research in this area. We also are very interested in individuals with idiopathic autism that have obesity and hyperphagia. Hyperphagia means, eating all the time. Sometimes all of us have a little bit of that, but this is a lack of satiation after a meal. So CFIP definitely is an important gene for appetite regulation too. So it's going to be interesting to understand how this is related to autism and whether CFIP downregulation can occur in other idiopathic causes of autism. I just want to show you a picture of the Fragile X gene. At the front end of the gene, there is a CGG-repeat. DNA is like a twisted ladder, and the rungs on the ladder have the nucleotides. So on one side of the ladder, there's a CGG repetitive sequence. Now, this gene has been very important for evolution of human intellect, and probably in evolution there turned out to be a greater number of CGG repeats because this can upregulate the expression of this gene with the greater number of CGG repeats. And this gene is so important for synaptic plasticity that in the evolution of human intelligence, this gene has been very important. But when the CGG repeat gets too big, like over 200 repeats, and this is the full mutation, it has a big problem with replication, and the body will methylate this gene or turn it off. And this is an example here. These little orange balls on the backbone of this gene is a methylation process, a CH3 process that silences the gene so that it doesn't produce any message or very little message. And here's the messenger RNA that's produced by the gene. And the message, if you remember your, genetics from high school, the message is what goes out into the cytoplasm of the cell and gets translated into the protein. Okay? So the lack of message and the lack of protein is what causes Fragile X syndrome. Now I'm going to talk to you about what happens in the carrier state or the premutation state, where there's 55 to 200 CGG repeats. Actually, compared to normals, this premutation produces extra messenger RNA. It can produce anywhere from 2 to 8 times normal levels of message. So it's actually just the opposite of Fragile X syndrome. Fragile X syndrome, no message, no protein. Fragile X premutation disorders, extra message. Okay? Now, usually the protein is normal or close to normal, but there are certain conditions that occur. Premature ovarian failure or primary ovarian insufficiency is where women can go into menopause or a menopause-like state without menstruating before age 40. And the premutation is the most common genetic cause of this problem. And there's also what we call the Fragile X-associated tremor ataxia syndrome, or FXTAS. And that's FXTAS. And I'm going to talk to you a little bit about this because the premutation is actually pretty common in the general population. Anywhere from 1 in 130 to 1 in 250 females in the general population has the premutation. And in males, it's anywhere from 1 in 250 to 1 in 800. So it's less common in the males, but definitely there's a lot of people in your area here that have the premutation. It's more common than individuals with Fragile X syndrome, which is about 1 in 2,500 to 1 in 3,600 in the general population. So these premutation-associated disorders are common. Now, why do they have problems? And it's because the premutation, or all of this excess messenger RNA, causes dysregulation of some of the proteins in the cell. In the neuron, the cell with the premutation divides more rapidly. So we see lots of individuals with the premutation that are extra smart, okay? But the neuron can die more easily with stress, okay, and with toxins too. So we're studying that too. We even see particularly young boys with the premutation who can present with autism spectrum disorders. And I'll show you about that. So when we think about, this is looking at the whole CGG-repeat range. Here's normal, here's gray zone, and actually in the gray zone between 45 and 55, this is a gray area of research because NIH hasn't funded it yet, but we see problems with the gray zone. The messenger RNA is 2 times normal. The rate of premature ovarian failure is 2 times the rate in the general population, and we're very interested in neurological problems and even autism spectrum disorders in the gray zone. But definitely I've had enough difficulty in this field just convincing people that premutation carriers can have problems. Everybody thought I was nuts for years, but then when we discovered the Fragile X-associated tremor ataxia syndrome, or FXTAS, you know, when you have a hard neurological problem that people are really suffering from, then you can't deny these difficulties. So that is really clearly associated with the premutation, along with premature ovarian failure. And we think there's psychopathology and other difficulties that sometimes can occur that we'll talk more about. But then when you go into the full mutation, the level of messenger RNA goes down when there's complete methylation. Sometimes it can stay a little bit elevated when there's a lack of methylation or a mosaic pattern. Some individuals, individuals that can have some cells with the premutation and some cells with the full mutation. And then the level of the Fragile X protein is normal for most individuals with the premutation, but it can go slightly down in the upper end of the premutation, and then it basically disappears when you have a full mutation male, but it can stay at a higher level in a full mutation female. So these are the kinds of problems we see. So there's a whole spectrum of involvement, whether you're talking about Fragile X syndrome, which means that this person has the full mutation, or Fragile X-associated disorders for all of these other problems that can be related to the premutation. And it was really the mothers who were talking to me about their kids with the full mutation. And here's 2 very high-functioning kids with the full mutation. This boy presents with Asperger's syndrome and full mutation. This girl who has a full mutation, who has done very well, she actually has a gifted IQ, but auditory processing problems and anxiety problems related to her full mutation. And this mother who raised these 2 Fragile X kids who have done very well also got the gene from her father, this first gentleman who we diagnosed with FXTAS, who was an electrician and developed an intention tremor, and this dramatically interfered with his job. And then here's a woman that died just last year. She had 10 children, several of them with the full mutation, many with the premutation, and many, many grandchildren with Fragile X syndrome. Followed at Kaiser for many years. She had years of fibromyalgia, years of anxiety, years of chronic pain and neuropathy. Actually became addicted to pain medication because of the neuropathy in her legs, swollen legs, as you can see here, and then developed tremor and ataxia. As you can see, she's in a wheelchair here, and so eventually was diagnosed with FXTAS. But we think that there is an association between the premutation and some autoimmune problems, including fibromyalgia and hypothyroidism, and I'll show you some of the data. Here's a mom. I don't know if you guys saw the Time magazine article that came out in July, but this family was highlighted because this mom has two boys. One has Fragile X syndrome, and it's this one who has slightly prominent ears. She herself has had ovarian problems and was diagnosed with POI recently, and her father has had very severe FXTAS. So there's multiple individuals. Actually, I should show these three kids, all with the full mutation. None of them presented with mental retardation. This one with shyness, social anxiety. This one with a big head and hypotonia, who has normal IQ and some language delays a few years later. And this boy, very severe hyperactivity, temper outbursts. He has 200 repeats right between the pre and the full mutation. Even though his IQ is normal, very significant problems with severe ADHD and mood instability and some paranoid ideation. So very interested psychiatric profile. Now, FMRP, as I mentioned, is this mother protein that binds with messages from many other genes, hauls these messages out to the synapse, and then it regulates the translation of these messages at the synapse. And I think understanding that FMRP is this really important regulatory protein that usually is inhibiting translation. So it's keeping things under control at the synapse, and only when the right kind of stimulation comes in does it de-repress the translation mechanism, and the right kind of stimuli comes in, and proteins are produced that strengthen the synaptic connection. So many other genes that we know are associated with autism are regulated by FMRP. Neuroligands, neurorexins, the ARC protein, MAP1 kinase, PSD95, MMP9, or this matrix metalloproteinase 9, which is going to be related to a new targeted treatment that I'm going to tell you about, CAM kinase, many other proteins. It actually, the lack of FMRP downregulates PTEN, and it upregulates mTOR. And for those of you who are really good molecular biologists, which there's probably very few in this audience, but that is meaningful for you. So here's a really complicated picture I just got at the Banbury meeting from Jennifer Darnell, a great molecular biologist. And this is the synapse. You know, this is the presynaptic part of the synapse, and this is the postsynaptic part of the synapse. And all of these proteins are really important for building and strengthening the synapse. And so she's done a lot of work about what proteins bind and are regulated to FMRP, and all of the boxed proteins here are regulated by FMRP presynaptically, and these boxed proteins are all regulated to FMRP postsynaptically. You know, most of you who know about neuroligands and neurorexins would maybe be interested in this aspect. But so we have a lot more to learn about what is controlled by FMRP. Now, we and others have reported about autism in premutation carriers. So in the past, when pediatricians or neurologists who order the DNA test and saw the premutation, you know, people used to think that the premutation was not associated with any clinical involvement, and so they would just ignore a premutation finding. But individuals with the premutation can look very normal. This little girl, though, has lots of features of Fragile X, hand flapping, hand biting, but lo and behold, she got the premutation from her dad, and she was diagnosed with autism years before she was diagnosed with the premutation, but she actually has lowered protein, as does this boy. Both of these individuals are intellectually impaired, but we've seen more recently a group of individuals with the premutation who have very high IQ, very high verbal IQ, who have very bright individuals who have more of an Asperger-like phenotype and meet diagnostic criteria for Asperger's with high IQ related to the premutation. So we were very interested in how often this happens. You know, when you see a kid in clinic, that's a very skewed sample. And so we compared in this study that was published in 2006, boys that presented to our clinic as the proband, found to have the premutation. Then we do cascade testing through the family. That means cascade, you know, do Fragile X DNA testing in other family members who are at risk to have the mutation. So we looked at the brothers who were the non-probands but found to have the premutation compared to the control brothers. So here's a boy that presented to our clinic at age 7 and 6. The proband presented with autism and ADHD. He was found to have the premutation. His brother here was also tested and found to have the premutation, but he has clinical significant anxiety and ADHD. So ADHD, very common in the probands, of course, because they're skewed, because they come into clinic. But cascade testing, non-probands had a pretty good rate of ADHD and different compared to their brothers. But autism spectrum disorders, 73% of the probands had an ASD. 4 out of the 14 had full autism. 7 out of the 14 had PDDNOS. Compared to the non-probands, we found 1 with full autism. But typically the non-probands had more shyness, social anxiety, social deficits, the SCQ, which is another faster checklist for social deficits, was significantly different than the normal brothers. So most of the time you get more subclinical or milder problems. Now we have looked at adults with the premutation in functional MRI studies, and these are adults who do not have neurological problems. And we find that the connectivity to the amygdala is problematic because the amygdala, or this emotional area of the brain, actually doesn't light up. Here's controls, and here's the young adult premutation group. The amygdala doesn't light up as it should to fearful faces. So there does seem to be some connectivity problem. You know, sometimes we can see older adults with the premutation that have social deficits or a little bit on the obsessive-compulsive side. Women with the premutation, particularly if they have more than 100 repeats, have even higher rates of depression. We've seen that anxiety, different types of psychopathology can, particularly obsessive-compulsive disorder, correlate with the level of messenger RNA. So, and this is particularly true in males, as we see here, premutation females and premutation males with obsessive-compulsive disorder, and this correlated with the level of messenger RNA in the males. So again, individuals with the premutation can be very bright, particularly in verbal areas. They sometimes can have anxiety, be a little on the obsessive-compulsive side, and we find, and this again may be skewed because you know, we bring in a number of people, and of the individuals that we've seen with FXTAS, we've seen that 40% either have a master's degree, a PhD, or an MD, or are lawyers, you know, Fortune 500 company execs. And I think it's because this phenotype of having good verbal abilities, being a little on the obsessive side, and kind of driven, you know, with a little OCD, gets you a long way in our society. Actually gets you to get higher education. So we've seen many individuals with the premutation that are on faculty. You know, that's a good place to screen for the premutation. On faculty, both at, you know, at University of Colorado and at UC Davis. So we've had to, because some people are so bright, we had to, for controls, we've had to get emeritus faculty just to match the bright IQ. Anyway, and I think this is because the premutation neuron replicates faster, and so that's part of why I think we see some with a higher IQ. But there are some individuals, particularly at the upper end of the premutation, who may have not only high message but slightly lowered protein. Isaac Pessa is a great neurobiologist associated with the Mind Institute, and he and Yu Sui Shen have looked at neuronal cultures with premutation neurons. And what you can get, and these are individuals that have, I think these individuals have around 150 repeats, but we can see differences, I'm sorry, this is 172 repeats. So we can see differences in the neuronal cell culture in terms of the branching here, less branching. The synapses, actually there's reduced complexity, but the synaptic size is actually increased. And let me show you this. The synapse is increased in size related to the controls. So we think that for some, there can be even neurodevelopmental problems. So I'm going to show you this whole Fragile X family here, where these individuals came in to the clinic. You know, this boy with Fragile X and autism, this girl with more milder involvement. The 38-year-old mother here has premature ovarian failure, anxiety, neuropathy, muscle pain. She was eventually diagnosed with lupus. Her mother here, up one generation, has tremor and ataxia, FXTAS, and had a history of early ovarian failure. And then her father, the great-grandfather of these kids, has FXTAS and cognitive decline. So he actually has dementia along with neuropathy. So we study all individuals in the family. So this is a very busy slide, but I'm trying to show some overlap in terms of pathogenesis for all these different causes of autism spectrum disorder, tuberous sclerosis, Fragile X syndrome, and Fragile X premutation, 15q duplication, MECP2 mutations, differences in serotonin transmitter, and there's a lot of different processes that can go on, all leading to, you know, either abnormal synaptic connectivity, GABA glutamate imbalances, CNS growth, and reactivity changes. So there's a lot of cross overlap in terms of what causes cell death, what interferes with synaptic plasticity, altered expression, etc. I think I better, let me just continue just a little bit. I'd like to show this picture because this is another cause of autism, PKU. Both of these girls have the same mutation. This one got early intervention because of newborn screening for PKU. And she has normal IQ and is marrying on this day, but her older sister was born before newborn screening was able to detect this mutation. So this is one cause of autism that is curable by diet. And, you know, we've known this for a long time. And, you know, I think people in the autism field would like a nice cure like this for all causes of autism. And of course, we do, you know, wonderful treatment. You know, I'm from University of Colorado, and Sally Rogers is one of my best friends, so I'm very instilled in the Denver model. You know, all of these variations on ABA intervention. Pivotal response training, I think, is very good too, from the Kegels. And, you know, all of these intervention programs, Sally Rogers has demonstrated really good efficacy in her intervention. It's been shown in other behavioral interventions for autism. You know, I think these interventions are wonderful. For multimodality treatment for Fragile X, Sally Rogers' intervention, I think, works really well for Fragile X, whether they have autism or not. We do a lot of combining speech and language and OT therapy, prompt therapy, this prompt intervention developed by Deborah Hayden and others. I think Sally Rogers has published on the intensive prompt intervention, where there's a lot of tactile, you know, input to the mouth to really stimulate language. I think is an excellent intervention, and it's available online, and we do a lot of programs at our summer institute at the Mind to teach therapists about this. There is a really nice website for the National Fragile X Foundation that has a whole section on special education intervention. I'm particularly interested in computer learning programs and programs that can enhance written language expression. But kids with Fragile X love computers and do very well on computers. We'll talk a little bit more about psychopharmacological intervention, but I want to spend some time, and since I only have a half an hour left, talking about targeted treatments for Fragile X that I think can impact other causes of autism too. So I will do a lot. The visual pathway for static visual pictures works very well in Fragile X. This is the ventral visual pathway in the brain. The dorsal visual pathway, that's more for visual motor processing and movement, is much more problematic in Fragile X. We do a lot of visual schedules, which of course are very helpful in autism. This is a little about prompt therapy. We're very interested in assistive technology devices, particularly for the 10% of kids with Fragile X that are nonverbal, and these communication devices. We work with Peter Mundy at the Mind, who's developing virtual reality paradigms for autism intervention, which we think is going to be really helpful for Fragile X intervention too. I think that you will see more and more about virtual reality paradigms. We're working with computer gaming people for early intervention for young children with Fragile X, and games that can improve visual, motor perception, executive function control. So we're excited about some of these computer learning games that can be started very early. Behavioral interventions. This is a family in South America showing me their best behavioral intervention for their wildly hyperactive kid with Fragile X, and other kinds of behavioral interventions, of course, calming techniques, not reinforcing negative behavior, timing out. I mean, these are standard behavioral interventions that work for any child with really severe hyperactivity and hyperarousal. I'm going to talk more about psychopharmacological interventions, but I think after the break, which I want to take now, I'm going to be talking about the new targeted treatments for Fragile X that I think can help autism too. And then if we have more time, we'll talk a little bit more about autoimmunity and FXTAS and premutation involvement. So let's stop for a break. I want to talk about targeted treatments, okay? I actually skipped all the sections that talk about stimulant medications and SSRIs, but I do want to say that mood stabilizers like risperidone and Abilify can be very helpful for individuals with autism, and also Abilify is probably the best medication out there that's available clinically for Fragile X syndrome, because it actually decreases anxiety along with stabilizing mood, decreasing aggressive behavior, and it just kind of helps in organized thinking and calming these individuals down. But I am really most excited about targeted treatments, and these are treatments that are related to understanding the neurobiological changes in the DNA in Fragile X and what that does at the synapse. I want to reiterate that FMRP, this red triangle, takes up messenger RNAs, probably from more than 100 genes, drags these messages down to the synapse, and it sits on these messages and regulates translation, as we've talked about. But one of the very important pathways that it regulates is called the mGluR5 pathway. And I apologize for being so molecular, but the only way you're going to understand these targeted treatments is to hear some about what makes synapses strong and what makes them weak. And the mGluR5 pathway, which stands for metabotropic glutamate receptor 5 pathway, so it's part of the glutamate system, is a pathway that leads to long-term depression. Now, LTD, or long-term depression, doesn't mean psychiatric depression. It means the process of weakening synaptic connections. So how do you weaken synaptic connections? Well, it actually is a process of pulling in receptors. And these glutamate receptors, called the AMPA receptors, are really important for learning and cognition. And when they get pulled in or internalized, it leads to a very weak synaptic connection. And this pathway, the mGluR5 pathway, leads to weak synaptic connection through this process. But guess what? FMRP is the main inhibitor of the mGluR5 pathway. So if you don't have FMRP, as in Fragile X syndrome, guess what? This pathway is upregulated. So there's weak synaptic connections all around the brain. So this was first discovered in the Fragile X mouse model by Kim Huber, working in Mark Baer's lab. And so this is a really exciting development because we can use mGluR5 antagonists to reverse this process. And guess what? These mGluR5 antagonists work really well in the animal model, okay, for Fragile X. They actually cure the cognitive problems, the seizures, et cetera, and I'll show that in a slide. But this slide just shows the difference between the knockout mouse and the wild-type mouse. And when the Fragile X protein is not there, many other proteins get upregulated because their inhibition is released. So there's a dramatic process of the development of weak, long, thin, and immature synaptic connections compared to normal synaptic connections in Fragile X. And what does this mean? When you study how the brain solves math problems in the MRI, and this is in girls here, this is a control group. When they're solving math problems in the MRI, lots of areas of the brain light up, so they're able to recruit a real big neural network to solve math problems. But here's Fragile X girls. These weak synaptic connections don't allow you to recruit the neural network you need to solve math problems. Okay? And that's the biggest academic deficit, even in normal IQ Fragile X syndrome patients, math. So we want to reverse this, and this is called the mGluR5 theory of Fragile X. There's an enhancement of mGluR5 activity related to the absence of the FMRP inhibitor, and this leads to enhanced long-term depression and these weak synaptic connections. It is what causes intellectual disability or mental retardation, and these connections are throughout the brain. If we use mGluR5 antagonists, we can improve behavior, seizures, cognition in the animal models. There's lots of evidence for this, whereas if you lowered FMRP, you pull in those AMPA receptors, and if you add back FMRP, those AMPA receptors go right back out and strengthen the synaptic connections. If you cross a Fragile X mouse with an mGluR5 deficient mouse, you cure Fragile X syndrome. That's pretty exciting. The only thing you don't cure is the macroorchidism, or the big testicles, but that's okay. My usual line is we think of that as a strength in Fragile X. Okay, so the mGluR5 antagonists, MPEP is the one that's been used in the animal models, but that's too toxic to use in humans. But there are many other mGluR5 antagonists. Phenobom is one that we have used in humans. Lithium has some effect on the mGluR5 system, and actually lithium is a really good medication in Fragile X. But Phenobom, we did a trial of Phenobom, and it was just great in patients. It was a single dose, And just after a single dose, you could see dramatic improvement in the hyperactivity, the anxiety, in the eye contact, and it was a very exciting day when we did our first phenobomb trial in Fragile X. But there are many other mGlu5 antagonists that are coming online. There's one being developed by Seaside Therapeutics. Novartis has one. Roche has one. The Novartis one has been tested in Europe, and it is good in adult Fragile X patients, and it's going to come to this country. And Roche has another mGlu5 antagonist that we hope to start trials in adults with Fragile X in September. There's a medication called R-baclofen, which is a racemic form of baclofen that is actually a GABA agonist that can downregulate the mGlu5 system. And we're studying that right now in individuals 12 and older with Fragile X syndrome. And R-baclofen is supposed to be very good in autism without Fragile X. And so Seaside Therapeutics is starting an autism trial very soon also. Now, minocycline I want to talk about because this is an antibiotic that lowers MMP9. But before I talk about that, let me show you just this R-baclofen structure. And R-baclofen is 15 times more potent than regular baclofen, and that's why R-baclofen is FDA approved, but only for research studies right now. There will be other medications that can upregulate the GABA receptor because that is downregulated in Fragile X. Ganaxolone is one that we hope to try in the future. This is just the structure of ganaxolone. But let me get to the minocycline studies. So the absence of FMRP. Remember how I talked about a lot of proteins get upregulated without the Fragile X protein? Well, MMP9 is another protein that gets upregulated. This protein is also really important for synaptic structure. It is probably ultimately regulated by mGlu-R, but it's an example of another protein that gets upregulated. It is definitely elevated in Fragile X syndrome. And minocycline, an antibiotic, a tetracycline antibiotic used to treat a variety of infections, and also used, it's a great treatment for acne in adolescents. It's probably most commonly used to treat acne in adolescents. But minocycline happens to lower MMP9. It also has some effects on the immune system, and it changes around cytokine and chemokine profiles. It has been helpful for some neurodegenerative syndromes, and we're actually going to be studying it in FXTAS soon. And actually, some of the other mGlu5 antagonists can be helpful for Parkinson's disease. And so we're excited to see as to whether it could be helpful. Some of these other mGlu5 antagonists could be helpful in FXTAS too. So some of these new medications have a lot of cross benefit to other disorders. But anyway, there is a minocycline trial going on now in autism. Sue Sweeto is doing that. But minocycline is a medication that can cause graying of the teeth when it's used in children that don't have their front teeth in yet. And so that's one of the reasons why it's not used in young children. But in the Fragile X knockout mouse, if you give minocycline for 1 month after birth, it normalizes synaptic connections. And that has some carryover even a few months down the line. Now, they saw improvement in the Fragile X knockout mouse in an anxiety and in also some cognitive tasks on the Y maze. So this is very exciting that minocycline could be a targeted treatment in Fragile X. Again, there was just a paper that came out about antioxidants, including alpha tocopherol, which is vitamin E, N-acetylcarnitine, and folic acid is also an antioxidant, that this may be very important in the Fragile X mouse model, but also in patients. It just hasn't been studied a lot in patients. Folate has been studied a little bit, but and it has some behavioral effects. But so there's going to be more studies done there. But let me just show you. I think I have a survey here about minocycline. This just shows MMP9 is a very important protein for the development of the synapse. In the Fragile X knockout mouse, which is here, compared to the wild-type mouse, and here's the Fragile X knockout mouse treated with minocycline. But the synaptic dendritic spines here are normal here, and they're long and thin and immature here, and they're improved here, but it's a little bit difficult to see, so let me just show you the quantitative thing here. In the wild-type mouse, the mature dendritic spines, which are the mushroom-shaped spines, they're fat and mature, compared to the long, thin filopodia, which are immature. The immature spines are much more so in Fragile X compared to the wild type, and the mature spines are underdeveloped, but when you add minocycline, it brings it back to the normal state. So this was published online in October, and a lot of families jumped on it and wanted to try minocycline right away. So just to give you some background, it was in 1963 that the FDA and the ADA acknowledged that you have these dental problems with minocycline used in early childhood because the tetracyclines actually chelate calcium and incorporate them into the bones and teeth. Minocycline was introduced in 1967. It has less phototoxicity compared to regular tetracyclines, greater GI absorption, it penetrates the CNS, less calcium binding, so less frequent tooth staining, and it has anti-inflammatory and neuroprotective properties. Overall dental staining after about a month's worth of minocycline only occurs in up to 6%, but it can also stain other tissues. Oxidation tends to bring out the dark colors, so maybe treatment with vitamin C at the same time may reduce oxidation. Minocycline sometimes can cause increased intracranial pressure. We call that pseudotumor cerebri, so we discontinue it if headaches occur. Sometimes emesis or GI upset can be a problem, and sometimes you can get an allergic or lupus-like rash with long-term minocycline treatment. So this just shows what the grain of the teeth looks like, but you can treat that with dental plating. Here's some dark coloration that happened with long-term minocycline treatment, but again, you can correct that with dental plating. Here's some more grain of the teeth. I'm not sure how well that shows up there. So a lot of families are thinking, well, what's it going to be? Good synaptic structure or gray teeth, you know? Basically comes down to that. One very important issue is that, of course, calcification of deciduous teeth is going on in young ages, even in utero, but the permanent teeth don't start to calcify until about 6 months, so there may be a window of opportunity in the first 6 months. And of course, minocycline is available by prescription. So we decided, because I've followed about 40 patients that have been on minocycline either from their primary healthcare providers or was started clinically by me or others, and so we decided to do a follow-up questionnaire. You know, 2 adolescents got headaches right away and went off, and 1 individual had diarrhea and vomiting and went off. So of the 37 who stayed on for at least 2 weeks, you know, about 50% were on the Autism Spectrum Disorder. Average age about 12 years, and minocycline mean treatment was 3 months, but 2 weeks to 12 months. So it did seem to, we actually divided the data between those treated less than 7 years of age, and the blue bars are those who are older than 7, and we only had about 7 children. But actually, a lot of them improved their language. And again, this could all be a placebo effect, and so I'm just showing you this data so that I think this data tells me that we have to do a controlled study of minocycline, particularly in young children, because young children, I think, have the biggest effect. In some, academic abilities were thought to be better, but in several, there was a worsening of the hyperactivity. So there can be some downside of minocycline. And inattention did improve, and focusing. Impulsivity did not improve. Overall, individuals less than 7 years of age, most of them had some degree of improvement from the parents' perspective. Those older than 7, there were some subtle improvements in many of these areas. And for those of you who might have older children that were treated with minocycline, I think there is some benefit and certainly worth a controlled trial. So that is one targeted treatment that is available now. And the others, again, we're going to do a phenobomb study in adults that will be a longer-term study. The Novartis agent and the Roche agent are coming this year for studies, at least in adults. I think we'll do a controlled trial of minocycline in younger children. We're particularly interested in babies that we diagnose in the newborn period because we have a newborn screening program now going. So we're particularly interested in all of the, much more intensive interventions we can give to the babies diagnosed with Fragile X very early on. And so we may actually do a controlled trial of 1 month of minocycline to see if it can improve synaptic connections. I wanted, because we have a number of carriers in the crowd, I wanted to show
actually just one hand. It's okay if you miss. That's okay.
In terms of the intention tremor in FXTAS. So this is an older male carrier. He's actually the first one that we diagnosed with FXTAS. So this intention tremor with finger to nose touching becomes much more dramatic. They can have problems in just pouring or eating, and ataxia, which is part of FXTAS, has to do with difficulty in tandem walking, heel to toe walking.
Okay, turn around.
And so individuals oftentimes can have problems with falling. Many of these individuals have Parkinsonian features, so often misdiagnosed as Parkinson's disease, autonomic dysfunction, either hypertension or orthostatic hypotension. Psychological features are very common, like anxiety, depression, or becoming more reclusive, and neuropathy. Painful neuropathy in the lower extremities is common, and on MRI we see white matter disease and what we call the middle cerebellar peduncle sign. We also see inclusions in neurons and astrocytes, and these inclusions can be throughout the brain in individuals where we've done neuropathological studies after death related to FXTAS. The inclusions are most dense in the hippocampus and in the amygdala. And so we think that they are caused by too much message binding with other proteins and pulling these other proteins into inclusions. There's a variety of dysregulation of other proteins that can lead to early neuronal cell death. Premature ovarian failure or primary ovarian insufficiency occurs in about 20% of women with the premutation, and an additional 23% can have menopause before age 45. So it just really shifts the curve down about when menopause occurs, but it also can cause an elevation of FSH, follicle stimulating hormone, even in women that are normally cycling, but definitely as they move into menopause, they can get an elevation of FSH. I wanted to just talk about a medical history that we did on 146 women with the premutation between the ages of 20 and 84 compared to controls. And again, this is a mom with the premutation with her child with Fragile X syndrome. So we divided those. 18 of them had FXTAS, and we matched that with age-matched controls, and then non-FXTAS women with controls. Now, FXTAS only occurs in about 8 to 10% of women with the premutation. We also see actually a similar incidence of premature ovarian failure, whether you have FXTAS or non-FXTAS. So again, about 20% here, only 13% here. So having premature ovarian failure does not mean that you will go on to have FXTAS, although I think both disorders are related to RNA toxicity. I think in primary ovarian failure, the granulosa cells that support the ovum, they die off, and that's why you can get ovarian failure, because it needs those support cells. Thyroid dysfunction, 50%, and it's usually hypothyroidism of individuals with FXTAS, 50% have thyroid dysfunction, significantly different than controls. Hypertension in over 60%, and hypertension is very significantly different than controls who are age matched, so we know it's part of the RNA toxicity process. Fibromyalgia in 43%, definitely different than controls. So some of these pain syndromes, muscle pain that hasn't been diagnosed as fibromyalgia, that is in 76%. So pain syndromes, not only neuropathy pain, but muscle pain and fibromyalgia definitely can be a problem, particularly in older female carriers. And females, of course, are much more predisposed to autoimmune disease than males. Later on this week, I'm going to Switzerland to talk about, to talk with the Roche people at a conference on Fragile X and autism, but they're going to present data on these mGlu5 antagonists, which they feel are very helpful for chronic pain syndromes. So the mGlu5 antagonists are helpful for migraine and chronic pain, and migraine is very much increased in individuals with the premutation also. So it could be that some of these chronic pain problems that we see in older carriers could be improved with mGlu5 antagonists. I just want to point out here, in this study, we had a few women that had MS, but because the numbers are small, it wasn't significantly different from controls. But we do see about 2 to 3% of women with the premutation develop MS. Now, I used to think it was just misdiagnosed and really was FXTAS. But here's a woman with the premutation who was diagnosed at 43 years of age with MS and died at age 52. And when she died, and she was diagnosed with the premutation just a couple of weeks before she died, but she had classic MRI findings of white matter disease typical for MS, and after death she had very active inflammatory lesions consistent with MS, but she had inclusions also all over her brain. So she had both FXTAS and MS. So FXTAS can occur together with other syndromes. So I just want to point out that tremor and ataxia are the main diagnostic criteria for FXTAS, but hypertension, psychological problems like anxiety, depression, migraine, fibromyalgia, urinary problems. We also see testosterone deficiency, hypothyroidism, autonomic dysfunction that can lead to impotence is very common among individuals with these neurological problems. Treatment for carriers, we always want to treat hypertension because hypertension going untreated leads to more white matter disease in the brain. We always want to treat the psychiatric problems, decreasing anxiety and depression, because depression can lead right into cognitive decline. And fluoxetine or other SSRIs stimulates neurogenesis. And that's what you want to do in aging brains. You want to stimulate neurogenesis. The other thing that stimulates neurogenesis is exercise. And that is important for every one of you out there. So you exercise not only to feel better, but it stimulates more neurons in your brain, and everybody can use more neurons in your hippocampus, okay? Everybody. We want to avoid oxidative stress damage. We do recommend antioxidants, and I think we got to do a lot more research. Processes like general anesthesia are really traumatic and kill off neurons, and so avoid general anesthesia. We're very interested in Neuroprotection, lithium. We're looking at memantine or Namenda. We're doing a controlled trial on Namenda right now in FXTAS, and we think it can be helpful at least initially. So also avoiding alcohol and other bad things you can do to your body that kill off neurons. Smoking kills off a lot of neurons. So all you smokers out there with the just stop smoking. So who to test? And the blood test for Fragile X is real easy. Every insurance company covers FMR1 DNA testing. Even state programs like Medi-Cal or Medicaid does it. Any child or adult with intellectual disabilities, mental retardation, autism, autism spectrum, or autistic-like features, all of those individuals should be tested. Someone with a borderline IQ or learning disabilities or ADHD and features of Fragile X, they should be tested. Women with reproductive or fertility problems, particularly those with elevated FSH, they should be tested. Those with late onset tremor, ataxia of unknown origin, I actually think we should be testing chronic pain clinics because I see that problem all the time. Multiple system atrophy, atypical Parkinson's disease, these kinds of neurological problems should be tested. We're doing newborn screening. I actually think very strongly that anxiety disorders, selective mutism, over-anxious disorders, there's a huge number of psychiatric populations that I think should be tested. Now I'm sensitive to the fact that the upper end of the premutation is, as FMRP goes down, mGlu-R5 goes up. And guess what? mGlu-R5 drives addiction and drinking, That's a whole nother field, addiction. And so I used to think that, a lot of old guys, particularly at the upper end of the premutation, that I see a lot of alcoholics in the upper end of the premutation, and I used to think they drank because they were anxious and maybe starting to have neurological problems, but now I think that it's mGlu-R5 elevations in the upper end of the premutation that's driving addiction problems. Neurological populations like multiple sclerosis, normal pressure hydrocephalus. Oh my God, before anybody does surgery on this, they should be tested for the premutation because I can't tell you how many guys with FXTAS have surgery for normal pressure hydrocephalus, which they don't have, and they just fall apart after that surgery, go downhill. We're doing newborn screening at our center and at Rush University, but OBGYN clinics, I think, should screen prenatally. And we're very interested in what is the best intervention for babies with Fragile X. So I'm writing a grant now about using Sally Rogers' early Denver model intervention combined with maybe some additional interventions for babies to see if we can really enhance their outcome, which I'm sure we can. You can get more information at fragilex.org about all kinds of, all the treatment centers that are around. There's 18 Fragile X clinics throughout the U.S. I'm sorry I didn't include a map, but there's not a clinic here. And there should be a Fragile X clinic here because all of, the Fragile X families, not all, but a lot of them come out to California to get, treatment and intervention. And I think the treatment should happen right here. You guys have a lot of expertise, and you just need to start testing and screening, and then I think you'll identify the individuals. And the Fragile X treatment centers will be included in many of these targeted treatment protocols, and you want all these good new mGlu-R5 antagonists here, not only for the Fragile X patients, but also for your autism patients. So I'm going to end there, and Amy's going to do some talking here. And, but I'll take questions at the end after Amy's lecture.